So hello friends, so myself Dr. Kishore Kunal and I teach botany in long-term Sri Chaitanya campus Gosala in both the boys campus and the girls campus. So today we are going to see a very interesting topic, and this topic is important at the same time because every year at least one questions come from this topic. So let's see this topic. So this topic is from the respiration in plants. And in respiration in plants, we will be majorly seeing the anaerobic pathway. Now, if we talk about the anaerobic pathway in the plants, so we already know that respiration is of two types: aerobic respiration, anaerobic respiration. The anaerobic respiration do not require oxygen. Aerobic respiration require oxygen. Aerobic respiration will also require mitochondria. Without mitochondria, aerobic respiration cannot take place in eukaryotes. So here, if I summarize this thing, so this is a overall summary of respiration. So as you can see, this the first step of respiration is the glycolysis. In glycolysis, the respiratory substrate, which is shown here as glucose, get converted into two molecules of pyruvate. Here no oxygen is required at all. So this is an anaerobic step. Now what we have got after glycolysis is two molecules of pyruvic acid. Now these two molecules of pyruvic acid can either enter into the anaerobic pathway or they can enter into the aerobic pathway. Now anaerobic pathway take place in the cytoplasm. So if it has to continue in the anaerobic pathway, it will remain in the cytoplasm, or if it has to undergo aerobic pathway, then it will enter inside the mitochondria, as you can see here. So this 2-pyruvate can enter into mitochondria and perform the aerobic respiration, or these 2-pyruvic acid can remain in the cytoplasm and perform the anaerobic respiration. Now this overall, these two steps together are called the anaerobic pathway. Now our aim in today's this video is to understand this anaerobic pathway. In this we will understand how glycolysis take place, how these pyruvate undergo the anaerobic pathway, that is through the fermentation, what are different types of fermentation, what are different steps of glycolysis, why is glycolysis important. Also, if you note, either in aerobic respiration or anaerobic respiration, in both this glycolysis is the common step. Aerobic respiration also starts from a anaerobic step, that is glycolysis. So glycolysis is common between both aerobic respiration and anaerobic respiration. So let's see one by one these anaerobic pathway. So if we see this, so this diagram is directly taken from NCERT. So as you can see, this is representing the glycolysis. This glycolysis, these are the different steps of glycolysis. How does glycolysis start? For anything to start, you need substrate. What is the substrate of glycolysis? So substrate of glycolysis, or you can say substrate of respiration, can be carbohydrate, protein or fat. All three can be the substrate, but most common substrate is carbohydrate. And in carbohydrate also, sucrose is the most common substrate for glycolysis. Sucrose is the most common substrate for glycolysis. This sucrose is you must be knowing, it is a disaccharide. Sucrose is a disaccharide which is made up of glucose and fructose. Now, if sucrose is the substrate, so this sucrose will get converted or break down into glucose and fructose using an enzyme which is called invertase, given in NCERT. The enzyme invertase will convert or will break down the sucrose into glucose and fructose. These glucose and fructose will then further act as the substrate in the glycolysis. As you can see, it is shown here in the NCERT diagram that it is starting with the glucose, right? So glucose either enter into the glycolysis, or the glycolysis can simply start with the fructose also. So if it starts with glucose, so glucose will get converted into glucose 6-phosphate, and then glucose 6-phosphate get converted into fructose 6-phosphate, or if it starts from fructose, then directly the fructose will get converted into fructose 6-phosphate. So these are the two entry points. Either fructose enters into the cycle through this pathway, or if it is glucose, then glucose through glucose 6-phosphate and then to fructose. So this one step of isomerism is absent if there is fructose as the substrate, and this step is present if glucose is the substrate. That is point number one. Now overall, if we see this pathway, it consists of 10 steps. If you count, it consists of 10 steps. Now these 10 steps are divided into two parts. One part is called the preparatory phase, another part is called the payoff phase. That we will see in a while. Let's first understand about the basics of this, from where the questions will be asked. So this glycolysis is called as the EMP pathway. Why it is called EMP pathway? Because it was discovered by three scientists, Embden, Meyerhoff and Parnas. From there, EMP pathway. Now this is the anaerobic step, so it will take place in the cytoplasm of the cell. Then it undergo partial oxidation. See, anaerobic respiration always undergo partial oxidation. It's only in the aerobic respiration that complete oxidation will take place. Now, what is the difference? How will you identify it is a partial oxidation or it is a complete oxidation? If it is a partial oxidation, then the product will not be CO2 and H2O. And if it is the complete oxidation, the product will be CO2 and H2O. So wherever you see that the product is CO2 and H2O, then it is and nothing else, only CO2 and H2O, then it is complete oxidation. But if there is anything else after still left, then it will be a partial oxidation. And you can clearly see the product of glycolysis is pyruvic acid, which is not CO2 H2O. So that is a partial oxidation. In this entire step, if you see, although it is a process to yield ATP, it will produce ATP, but in this process, 2 ATP is utilized also. That again we will see when we see the detail of these steps, right? So overall, if we see, then this glycolysis made up of 3 phases. So as I said, the first phase, which is called the preparatory phase. So let's see that phase. So this I have kept it as the preparatory phase. Here, as you can see, different enzymes are taking part in different steps of glycolysis. These enzymes, as you can see here, these are the enzymes which are taking part in different steps. So the first step, now let's assume that as it is in the maximum of the case that glycolysis start with glucose. So glucose is a 6 carbon compound. It is a 6 carbon compound and it get converted into glucose 6 phosphate. Means one phosphate is added at which position? 6th position. As you can see, this is the 6th position. Here one phosphate is added. So glucose to glucose 6 phosphate. And here the enzyme involved is hexokinase. Now this is very important. Many time question is asked that glucose converted to glucose 6 phosphate using which enzyme? Hexokinase. Fructose converted to fructose 6 phosphate using which enzyme? Hexokinase, right? So both, even if let's say fructose get converted to fructose 6 phosphate, still it is the hexokinase. Now here if you see ATP in this process, ATP is converted into ADP, means triphosphate is getting converted into biphosphate, means one phosphate is going where? To the glucose, right? So here first step ATP is used, right? So one ATP is used here. Here one more thing, the term kinase. Whenever you see the term kinase in the enzyme, it means either it is adding phosphate or it is removing phosphate. In the other steps also we will see wherever phosphate is added or removed, it will be the kinase. Clear? Now moving on to the second step. As I said, the isomerization step means glucose 6 phosphate. See, number of carbon is the same, phosphate is the same, it's just that glucose converted to fructose. Glucose and fructose are isomers. So this is isomerization. So now if I ask, glucose is used as a substrate and fructose is used as a substrate, what is the difference? The difference is the isomerization step. If fructose is used as the substrate, then isomerization step will be absent. But if glucose is the substrate, this isomerization step will be present. Now moving on to the third step. In this third step, fructose 6 phosphate means it has one phosphate getting converted into fructose 1,6. This 1,6 is representing the position of phosphate, means one phosphate is at first carbon, another phosphate is at the second carbon. So if you see 6 carbon and one phosphate at the first carbon, another phosphate at the 6 carbon. So that is fructose 1,6 biphosphate. Now here again you can see here only one phosphate, here second phosphate is added. So you can see another enzyme. See here also kinase. So now from here you can simply conclude wherever phosphate is being added and we will see removed also, it will be kinase. Clear? So kinase. Then coming to the, and since phosphate is added, so this step is called phosphorylation. Then this 6 carbon compound, which is fructose 1,6 biphosphate, is break down into two parts. One is glyceraldehyde 3 phosphate and dihydroxyacetone phosphate. It is with the help of an enzyme aldolase, right? So this breakdown, so now you can see the 6 carbon compound is now break down into 2 3 carbon compound. They both are again isomers. As you can see, 3 carbon 1 phosphate, 3 carbon 1 phosphate. So both are isomers. So here again the enzyme involved will be isomerase. So here again if I summarize here, see these two are isomers, so isomerase. Phosphate added, kinase, hexokinase. Phosphate added, phosphofructokinase. Again isomer, so isomerase, and here breakdown aldolase, right? So this we call it as the preparatory phase. Now in this preparatory phase, what you can see that one ATP is used here, another ATP is used here. This ATP is used as the source of phosphate. So 2 ATP are used. See, in this phase there is no production of ATP, no release of ATP, right? It is investing, means it is investing. So that's why it is called preparatory phase. So in the preparatory phase, 2 ATP is used. 2 ATP is used in the preparatory phase. Now moving on ahead, then we will see the payoff phase. So now the investment is done. Now it is time for the payoff. So already we have seen till this 5 steps, means the glyceraldehyde 3-phosphate and dihydroxyacetone phosphate are formed. Now this glyceraldehyde 3-phosphate, which is given in NCERT also as the PGAL, phosphoglyceraldehyde, 3-phosphoglyceraldehyde, right? So this then further get converted into, now everything has, so 6 carbon become 2 molecule of 3 carbon. So this entire payoff phase is multiplied by 2 because everything is 2 in number. All the product are 2 in number. So here what we see in the 6th step, that is oxidation and phosphorylation. See here 3 glyceraldehyde 3-phosphate and here 1 phosphate is added, 1,3-biphosphoglycerate. 1 phosphate is added, 1,3-biphosphoglycerate, right? Here, very important, 1 NADH is released. Now this is payoff, so production of ATP will start. This NADH will produce later. It will not directly produce ATP, it will indirectly produce ATP after in the aerobic respiration after oxidative phosphorylation. At this point we will not consider it as the ATP. It will produce ATP only in the aerobic respiration. Now in the next step, if we see 1,3-biphosphoglycerate converted to 3-phosphoglycerate. Here again you see phosphate is lost. So when phosphate is lost, again the enzyme is kinase, phosphoglycerate kinase. So phosphate is lost, kinase. Then 3-phosphoglycerate get converted into 2-phosphoglycerate. This is again a isomer. So here the word used is mutase. Mutase is also a type of isomer. Then 2-phosphoglycerate converted into phosphoenol pyruvate through with the help of an enzyme enolase, and here one H2O is released. Then in the last step what we observe, the phosphoenol pyruvate get converted into pyruvic acid or pyruvate. Here ATP is produced. Now since it is 2 in number, this entire thing, so here 2 NADH will be produced, here 2 ATP will be produced, here also 2 ATP will be produced. So here what we see in payoff phase, what we get 4 ATP released, we also get 2 NADH. This is what we got in the payoff phase, right? So, and in the overall, if we summarize, so in preparatory phase 2 ATP was consumed. So overall, see, these are the 10 steps. 5 steps goes into the preparatory phase where 2 ATP was used. So there we will write minus 2 ATP, and here 4 ATP is produced, so plus 4 ATP, and 2 NADH is produced, that is plus 2 NADH. Now moving ahead, if I summarize whatever we have seen, so in glycolysis, what we have seen in the preparatory phase, 2 ATP was consumed. This was in the preparatory phase. Then in the payoff phase, what we observed, we seen 4 ATP is produced and 2 NADH is also produced. This too is in the payoff phase. Now, let's see the energy balance. Now, as I said, this 2-NADH will produce ATP only in case of aerobic respiration, right? So, overall, here if we see net product of glycolysis. So, net product of glycolysis is 2 ATP and 2 NADH. 2 ATP and 2 NADH. But now here this product can go into two forms. Either it can go in aerobic respiration or it can go in anaerobic respiration. If it goes into aerobic respiration, then this 2 NADH will get converted into ATP, and here 2 NADH will get converted into 6 ATP. So 6 ATP from oxidative phosphorylation, and 2 ATP from direct production, that is called substrate level ATP. This is the substrate level ATP. Substrate level ATP. So this is only, we are now talking about the aerobic respiration. In aerobic respiration, the glycolysis, how many direct ATP or substrate level ATP is produced? 2 ATP is produced, which is substrate level. How many ATP is produced through oxidative phosphorylation? 6 ATP is produced during oxidative phosphorylation. How many total ATP produced in glycolysis in aerobic respiration? 6 plus 2. If they specify oxidative phosphorylation, you go for 6. If they say substrate level, if you go for 2. If they say total net ATP produced, 6 plus 2, 8 ATP, right? But what in case of anaerobic respiration? Anaerobic respiration, the case will be different. In anaerobic respiration, this NADH is not going to get converted into ATP. So here the ATP produced is only 2 ATP. So in anaerobic respiration, if the pathway is anaerobic, glucose will produce 2 ATP. If the pathway is aerobic, then glycolysis will produce 6 ATP through oxidative phosphorylation and 2 ATP through substrate level ATP. So that is total 8 ATP is produced in glycolysis if it is following the aerobic pathway, 2 ATP if it is following the anaerobic pathway. Clear? So now till this point everything is common. We have seen 2 pyruvate we have got. So in the end, as we can see here, we have got 2 pyruvate. Now this 2 pyruvate, as I said earlier, that it can either enter into aerobic pathway or it can go into the anaerobic pathway. So today here we are focusing on the anaerobic pathway. So let's see how it enters in the anaerobic pathway. So this is overall, this all step is the glycolysis, which has produced pyruvic acid. So this is the product of glycolysis. Now when it follows anaerobic pathway, so then that is called fermentation, which will continue to take place in the cytoplasm. And here, of course, the starting point again is pyruvic acid. Now, this pyruvic acid in fermentation can follow 2 pathways. Either it can form lactic acid or it can form ethanol, which is alcohol, and CO2. If it forms ethanol and CO2, it is called alcoholic fermentation. And if it follow lactic acid, if it forms lactic acid, then it is called lactic acid fermentation. Now, this of course take place in the cytoplasm. This will also involve enzyme. So here, in the formation of pyruvic acid to ethanol, see what is the difference in these two branch? You can see this here 2 arrow is given and here 1 arrow is given. So 2 arrow, why? It's not a mistake. 2 arrow means it is a two-step process. It is a two-step process. Conversion of pyruvic acid to ethanol is a two-step process. So it involves 2 enzymes. One will be called as pyruvate decarboxylase, which will help in release of CO2. And another enzyme used here will be ethanol or alcoholic dehydrogenase. This one will help in conversion of NADH into NAD+. Now see, NADH was produced in the glycolysis. In conversion of ethanol, see, in conversion of ethanol and CO2, the NADH is consumed. How many NADH is produced in glycolysis? 2. How many pyruvate is produced? 2. So 2 pyruvate will converted into 2 molecule of ethanol and CO2, and in this process 2 NADH will be used. So when I said NADH is not used to produce energy, means NADH is getting consumed. So in fermentation we see pyruvic acid get convert into ethanol, and in that process NADH is used. So the product of glycolysis was 2 NADH and 2 ATP. So 2 NADH is used. So what is the net product in this? 2 ATP, right? Similarly, if it follows the lactic acid pathway, here again the enzyme involved is lactate dehydrogenase. And this lactate dehydrogenase again get consume NADH. So 2 NADH which was present will again get consumed, and the net ATP produced will be 2 ATP, right? So here what we see, either it follows the ethanol fermentation pathway or it follows the lactic acid fermentation pathway. In both the case, how many ATP is produced? 2 ATP. In both the case, how many NADH is used? 2 NADH is consumed, right? So overall in the anaerobic pathway, whether it is alcoholic fermentation or whether it is the lactic acid fermentation, only 2 ATP is produced. That's why anaerobic pathway is not energy efficient. We will see that in aerobic pathway 36 ATP is produced, but in case of anaerobic respiration only 2 ATP is produced. So this is the contrasting difference between the lactic acid, between the aerobic respiration and the anaerobic respiration. So in every step, if I overall summarize it, so what we see here, if we see here, so 2 pyruvate is produced. If it follows the aerobic pathway, then it will require oxygen. If it follows anaerobic pathway, don't require oxygen. And here the net ATP produced will be 2 ATP, right? Here the entire pathway will take place in the cytoplasm, but for aerobic respiration part of it will be take place in the cytoplasm, that is glycolysis in cytoplasm, and rest of the reaction will be in the mitochondria. And this will further include two important steps, that is the Krebs cycle and ETS. So that we will see in a separate video. Let me know if you want, then I will explain this. So through comment you can recommend which topic here you will want to study further through the video. So this was all about the anaerobic pathway and respiration of plant in glycolysis. This is very important chapter, and one question every year definitely comes from here. So practice a lot of question from this topic, read from NCERT, no diagram will be outside NCERT, and you will do good if you revise it and just follow this video. So thank you all of you. Thank you very much.